Hi, this is Dr. Rosario, and welcome to our first video in our unit on microbial control and human health. In this module, we will focus on the control of microorganisms outside of the body. In our next module, we will look at the drugs that we use to control microorganisms inside the body. At the end of this unit, we are going to examine the different ways that microorganisms impact human health. We are constantly surrounded by microorganisms in our environment. Some of them can cause disease, while others spoil our food. A vast majority of microorganisms, however, do us no harm and are essential for our health and the health of the environment. In this module, we are going to start to look at the ways that we can control microbial growth to protect human health, while also considering when it is appropriate to use our control tools. In later videos, we will discuss specific physical and chemical methods for limiting microbial growth. Here in this video, we are going to discuss some of the general concepts surrounding microbial control and define some important terms. How do you know which microbial control method to use? Well, Stephen Covey once famously said, "Begin with the end in mind." Well, he wasn't talking about microbiology, but the idea is the same. What is the goal? What are we hoping to achieve here? There are four major outcomes of microbial control: sterilization, disinfection, antisepsis, and decontamination. These terms can often be confused, and sometimes students will use them interchangeably, but it's very important that you learn the distinctions between these four terms before you move on to our later videos in this module. Sterilization is the most extreme outcome of microbial control. It is the complete removal or destruction of microbial life from a surface or a substance. A substance that has been sterilized is described as sterile. It's the adjective there. It is important to note that a substance is either sterile or not. There is no in between. Sterilization is not appropriate for your hands or living cells. It is impossible to sterilize human tissue without destroying it. A substance that is used for sterilization, specifically a chemical that is used for sterilization, is called a sterilant. Sterilization is not always necessary. For example, the water you drink from the faucet or the plates you eat off of are not sterile, but they're usually clean enough that they won't transmit disease. On the other hand, critical medical devices must be sterilized. These are items that come into contact with blood or internal body tissues. So these are things like syringes, catheters, scalpels, and other surgical instruments. You should exercise extreme caution when using the word sterile. So students will often misuse this word in place of other more appropriate words to describe microbial control. Like I said, precision is important here. Sterile isn't just clean. Sterile means completely devoid of life. The process of disinfection inactivates most microorganisms, specifically pathogens on inanimate surfaces. A chemical that's used for disinfection is called a disinfectant. Disinfection is a very common process with wide applications, in contrast to sterilization. Choosing the right disinfectant depends on the circumstances. A semi-critical medical device is one that comes into contact with intact mucous membranes. Examples of semi-critical devices are endoscopes and respiratory therapy equipment. These items may not need to be completely sterile, but they are subjected to a high level of disinfection. Non-critical devices are items that are going to come into contact with skin or intact skin and not mucous membranes. These would be things like stethoscopes or blood pressure cuffs, even the bedding in a hospital room. Those would all be considered non-critical medical devices. When a nurse sprays down a table in an exam room with antimicrobial chemicals, this would be considered disinfection. Antisepsis is like disinfection, but it is specifically referring to the inactivation of microorganisms on living tissue. The act of wiping the skin with an alcohol pad before an injection is antisepsis. It can also be called degerming. An antiseptic is a chemical used to disinfect human tissue. Antiseptics must be carefully evaluated for safety before they are put to use. Finally, decontamination is the mechanical removal of most microbes from a surface. Mopping the floor removes the dirt and soil that may contain microorganisms. This is a type of decontamination. Washing your dishes is a type of decontamination. Decontamination usually involves the use of cleaning agents like soaps and detergents. Often, decontamination is paired with disinfection when antimicrobial chemicals are used. Asepsis means without contamination. Aseptic techniques in the laboratory or the clinical environment that prevent contamination. In the laboratory, we use Bunsen burners to sterilize our wire loops before transferring microorganisms from one culture to another. In the clinical environment, aseptic technique includes careful handling of patient specimens and infection prevention and control practices. For example, patients are draped with sterile materials and clinical staff wear appropriate personal protective equipment when performing surgery. When we think about different microbial control applications, we have to think about what is the context in which we are going to use them. So what is the function to sterilize or disinfect? Does it need to be sterile? Is it a critical medical device, a semi-critical medical device, or a non-critical medical device? How will it be used? Is it going to come into contact with patient skin, or is it something that's just going to be used in daily, day-to-day operations? Is an item going to be reused, right? In the medical environment, a lot of the equipment that we use is single-use equipment, and part of the reason for that is that it limits the potential for contamination. It limits the potential for the transmission of disease. Is the control method appropriate for the material that we are trying to either disinfect or sterilize? For example, you can't put plastic in an autoclave. An autoclave is a tool that we use that uses high heat and high pressure, but plastic will melt under those circumstances. Bleach is a chemical that we use for disinfection. It's great at removing microbial spores, particularly bacterial endospores from surfaces. It will inactivate those spores. Unfortunately, bleach also is an irritant, and it can damage surfaces, right? It can stain our clothes. It can be corrosive to certain surfaces. So it's not always appropriate to use bleach. You have to think about the context. One of the things we have to consider is the safety of our antimicrobial tools, particularly with our chemicals. Are there long-term health consequences for using certain chemicals in the healthcare environment or even in our homes? The other thing to consider is it cost efficient, right? Some of our microbial tools are great at inactivating or removing microorganisms, but they're not something that are going to be accessible to someone in their home or somebody just operating a small clinic, right? So you have to think about what is cost efficient and accessible. When we are evaluating different microbial control tools, you will commonly see two different suffixes used to describe those tools, either cytal or static. Cytal methods are methods that kill or inactivate microorganisms. So, for example, bactericidal kills bacteria. Virucidal inactivates viruses. Sporicidal is something that inactivates endospores, right? So in my previous example, bleach is sporicidal. Static methods, on the other hand, slow microbial growth. So for example, when we put our food in the refrigerator, that slows microbial growth, but it doesn't necessarily kill the microorganisms that are present in our food. That's why our food eventually will spoil, even if we keep it in the fridge. So microorganisms don't have lungs for breathing or hearts that beat. So how do we know if they are dead or inactivated? Ultimately, we measure microbial death by looking for the ability to grow or replicate in culture. This means using the same methods that are used for quantifying microbial growth. Most commonly are plate count methods. For each microbial control method, the rate of microbial death is constant. A percentage of the population dies or is inactivated over time. Like we see with microbial growth, plotting microbial death on a linear scale is not very useful. In this example, 99% of the bacteria are killed every minute. We started with 1 billion cells. Just looking at the graph, it appears that all the microorganisms are dead after just a minute, while in reality, 1% of 1 billion is still 10 million cells. Plenty of microorganisms still survive and can possibly cause disease. It is more informative to use a graph or to graph microbial death using a logarithmic scale, just like we do for measuring microbial growth. Here you can see that the death rate is constant and approaches 0 at around 5 minutes after exposure. There are many different factors that influence microbial death, the first of which is time. The number of cells that are killed is going to depend on the time of exposure to a particular method. Another factor that influences microbial death is the size of the starting population. Here we started with two different population sizes, one small and one large. The microbial death rate for this application is the same for both, and you can see that both of these lines have the same slope, but the time to sterilization for the line in orange, which is the one for our larger population, is longer, right? After 5 minutes with our larger population, there are still many organisms present in our sample. So the time to sterilize depends on our starting population. Some organisms are easier to eliminate than others. Prions and endospores are the most resistant to microbial control and the most difficult to remove from the environment. On the other end of the spectrum, most gram-positive bacteria and enveloped viruses are relatively easy to eliminate. This has important implications for infection control. Diseases caused by endospore formers, like Clostridium difficile, are incredibly difficult to control. However, those diseases that are caused by enveloped viruses can be easier to control, particularly in the environment. For example, HIV is an enveloped virus. That virus doesn't persist on surfaces for long periods of time, right? That has important implications for how the disease spreads. There are big differences in the way that cells growing in a biofilm methods compared to cells suspended in liquid growth media, aka planktonic cells. So here we have our biofilm and our planktonic cells. The biofilm resists penetration of antimicrobial chemicals, and it creates a unique microenvironment with lots of extracellular polysaccharides and proteins that interfere with antimicrobial activity. In addition, the cells within a biofilm often upregulate their stress response genes, and some of them enter a metabolically inactive form called a persister state. If you've ever tried to eliminate mildew growth from your shower, you know how hard it is to eliminate a biofilm. Another example of a biofilm that we encounter on a regular basis is plaque on our teeth. Again, that plaque keeps coming back every day if we don't constantly brush our teeth. Some other factors that we need to consider are things like the environment, right? The environment is going to play a role in the effectiveness of our antimicrobial treatments. For example, it's much easier to eliminate microbes from a stainless steel table than it is to remove them from carpeting. Temperature and pH can also influence the effectiveness of some of our disinfection techniques, particularly with our chemical disinfectants. Organic matter like blood, body fluids, vomit can infect disinfection. That is why cleaning is an essential step before disinfection or along with the disinfection process. The concentration or intensity of a microbial agent is an important factor to consider. One mistake that people often make is to dilute cleaning agents to make them last longer and to save some money. Unfortunately, this can undermine their effectiveness, and actually sometimes that can encourage microbial growth in those very same cleaning agents. Finally, the last thing to consider is the mode of action of a particular control method. There are different targets of microbial control. So a cell with a damaged cell wall is fragile and becomes easily lysed. Damage to the cell membrane results in loss of selective permeability. It also allows for vital molecules to leak out of the cells, and it allows damaging chemicals to enter in. Some of our targets of our microbial control pathways are going to be things like our biosynthesis pathways, right? So microbial cells depend on an orderly and continuous supply of proteins. So some of our substances inhibit ribosomes, which means they're going to inhibit protein synthesis. Some of our substances will damage the nucleic acids like DNA and RNA directly. Some of our substances or microbial control methods denature proteins. They disrupt protein shape, right? Some examples of that is moist heat. So we actually see this happen when we boil an egg, right? When we boil an egg, the protein in our eggs, that protein albumin, becomes irreversibly damaged as the proteins denature and they solidify, right? So that's an example of denaturation, disrupting protein shape in a permanent way. Strong organic acids and phenolic compounds also denature proteins. Some other things that can happen to proteins, so metal ions can bind to the active site of certain enzymes and prevent that enzyme from interacting with its substrate. So there's a lot of different targets of our microbial control methods. So this video was really just an overview of microbial control. In our next video, we are going to focus on our physical methods of microbial control.